Namaste and welcome back to Typical Monk. So far, we have seen the simple prokaryotic cell. Today, we step inside the eukaryotic cell. First, its skin, that is the membrane, and then its factory and post office, that is the endomembrane system. One idea at a time. Here is today's path. 5 stops. First, the fluid mosaic module of the membrane. Second, the plant cell wall. Third, the endoplasmic reticulum, that is rough and smooth ER. Fourth, the Golgi apparatus. And fifth, lysosomes and vacuoles. First, the cell's outermost living layer, the plasma membrane. The best way to understand it is the fluid mosaic model. This model was given in 1972 by Singer and Nicholson, and it is still the accepted one today. It has a double layer of phospholipids with proteins floating in it. These proteins are of two kinds: peripheral, which sit on the surface, and integral, which are buried inside. The membrane is quasi-fluid, that is, molecules can move within it, and it is selectively permeable, that is, it lets things in with care. Now, outside the membrane, plants have one more layer, the cell wall. Note this. The cell wall is found only in plants and fungi. It is not present in an animal cell. In plants, it is made mainly of cellulose, which gives the cell shape and protection. Its layers, in the middle lamella that join cells, then a primary and a secondary wall. Now, let's go inside the cell. The first big system is the endoplasmic reticulum. It is a network of tubes connected directly to the nuclear membrane, the factory floor of the cell. The rough ER has ribosomes on its surface. This is where protein is made and shipped onward. The smooth ER has no ribosomes. It makes lipids, that is fats and steroids. The ER, Golgi and lysosomes together form the endomembrane system, one connected network. Now, after the factory, the post office, the Golgi apparatus. These are stacks of flax sacs called cisternae, the post office of the cell. It has two phases, the cis, that is the forming phase, which receives, and the trans, that is the maturing phase, which ships out. Here proteins and lipids are modified, packed, and sent out with an address. And this is also where lysosomes are formed. Now, the sac that does the cleaning, the lysosome. It is a membrane-bound sac formed from the Golgi. It is full of hydrolytic enzymes that digest carbohydrates, proteins, fats, and nucleic acids, all of them. That is why it is called the cell's suicide bag. It can even digest the cell's own worn-out parts. Now the storehouse of the cell, the vacuole. It is a membrane-bound empty space. Its membrane is called the tonoblast. In a plant cell, it sometimes fills up to 90% of the volume. It stores water, ions, and waste, and keeps the cell firm. This is called turgor. Now, the high-yield pointers, the facts that NEET turns into questions. 1. The fluid mosaic model, Singer and Nicholson, 1972. The membrane is quasi-fluid and selectively permeable. 2. Rough ER makes protein. Smooth ER makes lipid and steroid. This matchup is asked a lot. 3. The Golgi has two phases, cis and trans. It does the packing and forms lysosomes. 4. The lysosome has hydrolytic enzymes and the vacuole's membrane is the tonoplast. 5. Animal cells have no cell wall. The plant wall is made of cellulose. Now let's clear the mistakes students fall for. Myth: rough ER makes lipid. Truth: rough ER makes protein. Lipid is made by smooth ER. Myth: animal cells also have a cell wall. Truth: only plants and fungi do. An animal cell has only a membrane. Now, test yourself. Think of each answer before it appears. Answer first. That is how you build strong NEET recall. Question 1. Who gave the fluid mosaic model? Answer: Singer and Nicholson. Question 2. Which ER makes protein? Rough ER. Question 3. From which organelle are lysosomes formed? from the Golgi apparatus. Question 4. What is the vacuoles membrane called? The tonoplast. Question 5. Which cells lack a cell wall? Animal cells. Let's gather it all in one breath. The membrane is a fluid mosaic, Singer and Nicholson, selectively permeable. The plant cell wall is cellulose. Animals have none. Rough ER makes protein, smooth ER makes lipid. The Golgi packs, ships, and forms lysosomes. The vacuole, tonoplast, storage, and turgor. In the next class, mitochondria, plastids, and the cytoskeleton. That is, the cell's power and its frame. See you there. You have understood both the factory and the post office of the cell. Be proud of that. One cell at a time with a calm mind. And you will surely clock NEET. This is Typical Monk. Every class, always free.